In this tutorial video, I will introduce SAED. This is a main component in the Landyne software suite for electron diffraction simulation and crystallography analysis. You may download the software suite from the website on the screen. Double-click this icon, you have the launcher set out. This one is the software we're going to use. Click one. If you have a license file, please copy the license key to the Landyne SAED folder. I'll show you in this PC, System C, and Landyne SAED program. In my case, I have the SAED3 and the SAED3 license key here. If you didn't have the license key now, you may select the demo mode when you start the program. Let me introduce the functions on the menu briefly. For Crystal, we have a new for you to prepare new data. And for load is load new data. Remove all means once you load multiple data there, you can clean out all of them. Exit is to leave the program. In experiments, you can load your diffraction pattern. We have another way to load diffraction pattern. Is used to drag and drop box. Invert is change the contrast of the diffraction pattern. Erase will clean out the diffraction pattern. You can do some process for your diffraction pattern, resize it, rotate it, or move it. This allows you to move your diffraction pattern, the incident beam, to the screen center. It's very important to do the calibration using this function. So it's called reciprocal space marker. This is a main function for you to find the zone axis of your experimental diffraction pattern. This one allows you to show the diffraction pattern name or not show the name. In simulation, we have two series for you to select. Normally, we select kinematic series calculation. And occasionally, for a small unit cell, you can select this dynamic series for calculation. This use a Bloch wave series. And you can change the microscope high tension, the high voltage here. Usually we put 200 kV there. You can set up the reciprocal limit here. And this is for the pattern zoom and the intensity scaling. And this is for you to put the index on your diffraction pattern. And this one is special for hexagonal phase. You can change it between two Miller-Brave index to Miller index. This is deal with the weak intensity special treatment. And this is to show you the lattice only. Sometimes you don't have the correct structure file, you only know the lattice. This function will be useful. In the pattern, first one is the region of interest and the TIFF. For you select how large area you want to print out. And in the printout, you can set up a resolution for the TIFF. And you can select the display as JPEG file or TIFF file. In option, we have a look and feel. You have 3 choices here. And for hide the toolbar for the graphic menu, or you can bring the graphic menu back. In the table, we have a elemental periodic table here. And we also have the space group table here. In the help, we have the current drive here. For my case, now it's a C drive. This is serial number. If you apply a license, you copy this number or take a snapshot of the whole window here and send to us so we can prepare your license file according to the serial number here. The last one is version. The current version is SAED to 3F, and you can find out the update date is here. If we upgrade the software, you find out maybe other letter is here. They're all version 3. The graphic menu bar here are part of the drop-down menu, but it is most often used one, at least here, except for the drop box. It's only appeared in the graphic menu here. Let's make the SAED pattern simulation. First thing is load a crystal. Let's try the aluminum. If you don't have the license, you may use the aluminum default. The default setting for the simulation is kinematic theory, and the max scope high tension is 200 kV. And you can see here the default aluminum here. If you load a crystal here, you will have one more file here. You can select the current file as aluminum here, or you can select current file as aluminum default. The following calculation will always use the current file. Simulation is a simple click calculate. You got a diffraction pattern here. Now we may change some parameters. First, the thickness. Default is 300 angstrom. Let's put 100 angstrom here. Do calculation. and you see more diffraction spot come out. Second thing, let's try to change the zoom axis, and we change it to 0, 1, 1 direction. See different diffraction pattern come out, and change it again for the 1, 1, 1 direction. 6-fold pattern here. Let's go back to the 0, 0, 1. And now let's try to see if there are small tilting angle here. We put 5 here. You can see the pattern is shift to the right side. If I put another one here, for example, minus 0.5, see the pattern is the center moved to the right side, down right side. Now I change it to the zoom axis back to 1, 1, 1. I want to talk about the mass proportion factor here. This value change from 0 to 1. And this is individual phase, the phase you're working on. It would affect the whole overall intensity here. If you have a multiple phase diffraction pattern as a composite in the display, the final proportion should be normalized. Just sum all the individual phase proportion and use the individual one to divide the sum of that proportion. Another thing is this one or this one give you this window is a zoom pattern, the whole pattern, and the intensity is not one phase but all the phase in display. Let's try this. So you can adjust the pattern, the size you want. The intensity, let's try this. Another thing is the orientation here. All of the slide bar, you can change it directly, or you use the mouse point to the slide bar and using the arrow key more precisely to adjust. Now we adjust it to 90 degrees here. So the new appearance of the pattern is shown here. All the pattern can be appeared as a circle as a default, or you can select the disk and change the color to red, and you can change any color you want, and even the user select the color, and here show customized. Let's go back to the black one. That's gray. This is black. If you have a multiple phase, you can even show this one in square or triangle just to distinguish them. Let's go back to circle. The next one is index. If I select index here, it will give me the index of the pattern. There are one more control of the index is here. You can change the color. Now it's in gray, barely to see. You can put it right there. If it's too small, you can change this font size to 18 immediately to see it. And also you can change how many index show up. If you lower the intensity to all of them, and all pattern will be indexed here. Otherwise, you can select the most intense one, or you can only show this is the base of only 2. With this 2, all the pattern can be indexed. And we can show the base vector too. So remove the base vector, remove the base only. It's pretty empty on the side of the pattern. So you can turn the ROI on to show. So the pattern will be printed out just from this blue squares. If we change this one to 400, so only this area will be printed out. For TIFF file, the PPI usually required to 300 or up for a publication figure. So once this done, you can go to Save TIFF, and this is you can change the file name, but we keep it as SAED pattern TIFF. And let's check out. Go here. This is in the result. And we have so many files created before, but this is the one we created now. Double click it. You can show from the photo. That's the pattern you get is a TIFF file. SAED3 can also simulate twin electron diffraction patterns. And there are two kinds of twins. One is a rotation twin, and the other is mirror twins. And we can also simulating the composite pattern for two-phase have a fixed orientation. Now we go to a new topic to study the experimental SAED pattern. First, I'll show you how to load the experimental pattern. To go find your pattern first. The pattern is in C pattern, C folder, and then 3 experiment here, and this pattern we just drag and drop to this box. The pattern will automatically load it. We want to move the incident beam to the center of the screen, and they come to this one, the shift and center, or in the experiment shift and center. Move this one to here, and you can see you can only show circle, or you can only show the cross lines, and you can decide how many circles here and how many lines there. Now it's 8 line of them. So once you have this line or circle appeared, you can just point to the center, and also the mouse left button and drag the image and release. So hold on and drag them and release. And in that way you can see the symmetry spot all in the same circles. Try to see that. Reciprocal spacing marker. So you can show the circle here, and also we know like the first one I said is 2.95, and I selected, no, it is 2.95, and here I selected is 1 nanometers, and I update. This is 2. So it's 2.95 update here. So it's pretty small. It's now covered to distant to here. So then come to the calibration part, and the pattern size here. We had to zoom this one, or here. We had to zoom here. Over here. So this one will show 2.639, and the circle cover to here. So we got here, we measured is 2.95, 1 nanometers, and then we, after we zoom the pattern, use this value, we get the correct circle size. So we do the update here, and that's do the calibration. Here there will automatically 2 vector here, but we need to point out the nearest one, like this one and this one, for us to find out. Now first to the angle to this line, and we need to shrink the pattern. You can fill the certain value directly here, and this is 88. You can fill the 44. Click here again and increase smaller. Not necessarily this one is fixed, but you have all this line of the marker, so to fix to that line approximately. So increase. Try your best to fit in. Yeah, this will be my best to fit in. All the markers are filled in this line. So we find that the second line will be to this one. So then I decrease my angle. Increase a little bit. A little bit. And this value now is 100, is about 50 here. I can put 50 here. Increase. And now, since I need to decrease a little bit here. Now is the best fit for this line too. So we got to find the 2 basic vector here, and we also have this tolerance factor 5%, and then we got to find. We find is the zone X is 211, and I put into red, and now I can get rid of this calibration. More circles. Scale circle now show. because the pattern are not tilted correctly, I need to reorientate the pattern. And it seems that perfectly matched with pattern, with this pattern, with most of the intensity here, 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 and also the lattice also match well. I can put a disk here to show you, and a circle here to show you. So it's a perfect match, except this point, and it could be because the simulation just approximately to match the factor of this one. But it's good, this one also match good. So this zone axis must be 211 zone index, and show the index here, and we get the index we can show in blue, and we can get a big number here, 24. And now is only index the most intense one, and I can reduce the limitation of the intensity, and you get all the most intensity pattern are indexed. Yeah, so all the most intensity index. Or you just simply go to the basic one. So we got this one is indexed, and also this one is indexed. And inside of this one will be this reverse. And we got some number here, -1. And I can show you maybe in yellow or other colors will be. Let's try to go to yellow. See if this one will show you a little better with 0, 1, -1, and this is 1, -1, -1. Thank you for watching. Please check our other video about Landyne software suite for electron diffraction simulation and crystallographic analysis.